I'd like to welcome you to our webinar series about laboratory good practices from Cryopreservation. My name is Sven Mühlfriedel. I'm a product manager at Cryopreservation, and in our company, I'm responsible for cryogenic storage labware. In today's second event of our webinar series, I'm going to talk about the cryogenic storage of biological samples. In case you have any questions, please refer to the end of my presentation when contact information will be provided.
My presentation is divided into the three parts: basic of cell banking, contamination, and high-throughput cryostorage. In cryopreservation, ultra-low temperatures are applied to keep biological samples unaltered or to preserve the structure and function of cells and tissue. What usually happens is that ice is forming in your sample, and once frozen, cells and tissue can be stored in a very stable state. An important temperature here is minus 140 degrees Celsius, which is the glass transition point of water. We know today that virtually all life processes are standing still if we have this temperature or go below. Cryopreservation is applied in cell and bacteria banking, usually temperatures ranging from minus 80 degrees down to minus 196 are applied. In biobanking and liquid biopsy banking, where the temperature range is identical, and also in tissue banking, where samples are stored at minus 80 or at minus 20 degrees Celsius. So let's now take a closer look at the basics of cell banking, where I want to provide several good practice tips. So I want to start with the workflow of cell banking, which is shown on this first slide. Everything starts with the preparation of samples. Afterwards, samples are frozen. They are stored, often transported, and everything ends with the thawing of samples when the cells are recovered and made available for culture or other subsequent analysis. The different stages of the workflow are associated with different challenges, and also important decisions have to be made. During the preparation of your samples, you want to make sure you choose an appropriate container system and also a good cryoprotectant agent. During freezing, you have to make sure that you work with an optimum freezing protocol, that you control freezing rates, and that you reduce osmotic damage which may occur during this step. During transport and storage, you want to make sure that your storage conditions are very carefully chosen, they are defined and maintained. You have to assure that a good system is in place for sample tracking, because you definitely don't want to use track of your samples. And also think about sample picking, because if this is not done carefully, some samples may be picked, others may remain in the store, and these other samples may be exposed to unwanted higher temperatures for long periods of time. During thawing, you want to make sure your samples are thawed very quickly, your CPA is eluted quickly. Thus, you can avoid and reduce chemical toxicity effects, and you round up everything by a viability assessment at the end. Thus, you can actually assess the quality of your recovered cells before you use them in cell culture or any other downstream applications. I want to focus now on sample preparation and its critical parameters. First one is cell concentration. It is most often the case that 1 million cells per milliliter are used. This is kind of a gold standard, and we know this is optimum for almost all applications. However, there are deviations from this gold standard. They go towards the direction of less concentrated vials, and this is often the case when your stock concept requires that you have more individual vials you can pick, and then of course this goes to the expense of the cell concentration. On the other hand side, there may be concerns that the viability of cells per vial is too low, and then you may actually decide to go for higher concentrations of cells to make sure that more viable cells can be retrieved from one individual vial. A good recommendation here, an upper end of the scale, is 10 to the 7 cells per milliliter. I already talked about cryoprotectants. These agents are protecting from damaging effects of freezing at low cooling rates. They depress the freezing point of the solution, and they also lead to less ice formation. They basically fall into two groups: the small molecular weight ones, which are penetrating the cells, the high molecular weight ones, which are non-penetrating. A famous or well-known example for the first group is DMSO, also glycerol, and in the second group we find sucrose as a very often and widely applied cryoprotectant. If we now think about sample volume, it's getting a bit more complicated. It's a lot of factors influencing here and leading to your final choice of a good sample volume. It may depend on intended cell concentrations and also your general stock concept. One recommendation we can, however, formulate, and this is think about a maximum size of a vial, because if you exceed a certain volume of sample which is frozen down, you may run into problems with cooling rates. In other words, large sample volumes are very hard to be controlled in terms of cooling rate. Last not least, there's a different group of additives added to samples during sample preparation, and these are excipients. They are actually there to improve cell viability, increase cell viability. Serum is a very well-known one, very often applied. If for some reasons, however, you want to be serum-free, then platelet lysate is an alternative. The two most commonly used cryoprotectants are DMSO and glycerol. On this fact sheet, they are quickly characterized, with DMSO lowering the freezing point, increasing dehydration of cells, reducing the intracellular ice formation and thus cell damage. DMSO being relatively harmless in concentrations below 10% and thus being used in concentrations from 5 to 10%. DMSO is usually sterilized by filtration through 0.2 micron PTFE syringe filters. On the other hand side, there's glycerol, which is reducing freezing artifacts. It's less toxic than DMSO. It's used in the same concentration range from 5 to 10%. It is sterilized by autoclaving because it's a very viscous solution, and it should be protected from light. On the right-hand side, you see a few recommendations for specific types of specimens. It starts with bacteria and yeast. For both of them, it's recommended to store them in 10% glycerol containing preparations. Protozoa are usually stored with 5 to 10% DMSO or 10 to 20% glycerol. Algae are stored in 5 to 10% DMSO, but sometimes also in methanol from 5 to 10%. Plant cells and animal cells can be stored in 5 to 10% DMSO or 5 to 10% glycerol. And hybridoma cells and stem cells can also be used in 5 to 10 DMSO containing preparations. It is interesting to note that for hybridoma cells, often serum is added at a concentration of 20%, and for stem cells, the concentration of serum being added ranges from 20 up to 90%. Before we move on with freezing, I want us to have a quick look at cryoprotectants again and come up with some best practice tips here. So the first one is use single volumes aliquots to reduce the risk of contamination. Second one is prepare everything freshly. In particular, this is important for growth media. They should be freshly prepared, then add your cryoprotectant, and afterwards add everything to your cell suspension. Further, each lot of cryoprotectant preparation should be tested for unwanted properties with a sensitive cell line. And last not least, go for the maximum, determine the highest amount and concentration of cryoprotectant still tolerated by your cells, and then use this in your preparation of cryoprotectant solutions. A very critical step in the workflow is freezing, because here a lot of adverse phenomena may occur. Of course, you have prepared for this because you have added cryoprotectants to your samples. However, I want to quickly go through the main adverse phenomena that may occur and want to give some background information here. First group of phenomena is described as solution effects. What is behind is the idea, the concept that when ice is forming, when water is freezing, solutes become excluded from the water phase and their concentration is increasing, and this simply can be dangerous to cells. What also may happen is that extracellular ice formation occurs. This is often the case when tissue is frozen down at a very slow cooling rate, and this finally leads to ice crystals damaging the cell membrane and leading to dehydration of cells. The dehydration is an as a single isolated phenomenon, describes the fact that the water is migrating out of the cells, maybe because of cell membrane damages, and this simply leads to cellular stress. It can cause damage to cells, it can reduce the viability of the cells, and can also lead to cell death. A phenomenon which almost in any case leads to cell death is intracellular ice formation. Intracellular ice is in almost any case lethal and cells are dying. So in order to fight all these adverse phenomena, you have to find an optimum. The optimum towards freezing has to do with an optimum cooling rate. The reason is indicated here. If you cool too quickly, then you have more intracellular ice formation. You will observe less osmotic imbalance. But I just told you how critical the intracellular ice formation is. On the other hand side, if you cool very slowly, you get a very nice control over intracellular ice formation. It's minimized, but you get more osmotic imbalance. So the optimum in between has very clearly been identified as 1 Kelvin cooling rate per minute. This is optimum for almost all cell types. There's basically 2 different types of devices available today for controlling freezing rates. On the left-hand side, we see a passive cooling device. Such a passive cooling device utilizes conducting media such as isopropanol or high-density insulating foam, and everything is incorporated into a heatsink device. When these devices are stored in a -80-degree environment, then you actually achieve, because of their design, a very close to 1 Kelvin per minute cooling rate. And all this is true for a temperature range from -10 degrees Celsius to -40 degrees. On the right-hand side, we see an example for a controlled rate freezer. They either utilize liquid nitrogen or a heat pump driven system. Here you can program very precise cooling profiles. You can even have step cooling profiles where you hold a certain temperature for some time. This is sometimes good and critical for very specific cell types or specimens. And they also are compatible with a very wide range of vials and bags. So you are very flexible in the choice of your storage container. Here you see a recommendation for a workflow for freezing down mammalian cells or cultured cells. First of all, you should harvest these cells from log or early stationary growth phases, and you should also remove anchorage-dependent cells with the cell scraper. You should prepare the cell suspension and your pre-sterilized cryoprotectant in twice the required final concentration, always in fresh media, as I already told you about earlier. And then you should mix 1 volume of your cell suspension with 1 volume of your cryoprotectant. Let everything equilibrate for 15 to 45 minutes, then aliquot into cryovials, cool and freeze as discussed earlier, and place everything in your storage. Some more recommendations on this slide. To start with stem cells. When you freeze stem cells, you should use DMSO plus 20 to 90% serum. This improves cell recovery and clonogenic activity. You may also consider to add trehalose and use a stepwise equilibration and a slow freezing rate. Also vitrification, a different approach we haven't really talked about so far, is a good choice sometimes for stem cells. If plant cells shall be stored and preserved, harvesting in the log phase is very critical. 2-step freezing is recommended, with the first step at -30 degrees Celsius, and then finally you go down to -150 degrees Celsius or lower. And freezing of seeds is of course an option if it comes to plants. Last not least, bacteria and yeast. They should be harvested from aerated cultures from late log or early stationary phases for optimal resistance to freezing processes. And you should use large numbers of cells per milliliter. 10 to the 7 or higher is recommended here. I want to move forward towards storage and want to come to storage containers. Different types of storage containers, the most commonly used ones are shown here. On the left-hand side, vitrification straws, in the center, cryovials, and cryobags to the right. Amongst the different types of storage containers I just showed to you, cryovials play a very important role. In other words, cryovials are very widely used in cryopreservation. With this slide, I want to draw your attention to several features such tubes can have so that you can make a good decision and a good choice. First of all, they come in different sizes and shapes, and it's good to make a good decision here. What is a good working volume for you? Even if they differ, most vendors offer the smaller volumes ranging from 250 to 500 microliters working volume. Then intermediate volumes ranging from 1 to 1.5 or 2 to 2.5 milliliters, and larger volumes in the range of 4 to 5 milliliter. There's also larger devices or containers available, but keep in mind that the larger your container is, the more you have difficulties with controlling freezing rates and finally achieve freezing rates that are good for your process and your sample. In the center portion, you see an example of a 2 milliliter tube with an external thread to the right hand side and internal thread to the left. It's a bit of philosophy what to use here, and it's also a lot depending on personal preferences. However, today we see a trend towards the internally threaded tubes when the intention is to maybe use automation and go for more automated processes, because here the internal thread of the tube, the external thread on the screw cap have proven to add process stability and automation compatibility. If on the other hand side, it's more manual steps in your workflow which need to be carried out, often the users, the scientists, technicians, they feel that the externally threaded tube adds more ease of use because the gripping surface on the screw cap is larger. On the right hand side, you see a rather classic tube to the left, which has a writing area where you can just write what about the content or the identity of the tube with a pen. On the right hand side, you see a similar tube or an identical tube, but it has a machine readable linear barcode and a data matrix code, and also a unique identifier which can be human readable because it's printed there on the side. We want to make you aware of these tubes, that they exist, because these barcoding options, and we will see more about it later on, can actually help in your sample tracking. They also add automation compatibility, and also note that the barcodes on these tubes can be customized. So for the storage of samples at very low temperatures in the range of -150 degrees Celsius to -196 degrees, liquid nitrogen tanks are available. Here the storage happens in the vapor phase above liquid nitrogen. Very often this is used for eukaryotic cells and protozoa. And what is important is make sure your vials are approved for storage at these low temperatures. The same type of specimen, eukaryotic cells and protozoa, can also be stored at ultra-low temperatures in ultra-low temperature freezers. These freezers use electricity to cool the air down, and they often come in the form of a chest freezer, because this is more economic. The very cold air is much heavier than the warm air, and it would not escape this type of freezer if you open the lid. In contrast, an upright freezer, which is often used for -70 or -80 degrees Celsius storage, often used for bacteria and fungi, provides better access to the samples, but you little bit lose the economic advantage of the chest freezer. Here you see 4 best practice tips for storage. The first one is about working volumes. You should never exceed the working volume as it is indicated by your vial supplier. And the idea behind is that usually samples are containing water, and water has its highest density at 4 degrees Celsius. In other words, when you go further down in temperature, the water is expanding, the ice is expanding. And the extra volume that you need for the expansion of your sample is actually taken into account, and that's why the working volume is usually less than the total inner lumen of your tube. It's about 90%. So keep this extra space available for the expansion of your sample, and if you do so, you don't have to be afraid that your tubes are rupturing or breaking and sample is leaking. You should also never store vials in liquid nitrogen, but only in the vapor phase above. The idea here is that if liquid nitrogen would enter your tube and you would recover your sample afterwards, then your liquid nitrogen, which has entered, would very quickly expand to an extreme extent. It would expand its volume 700 times. And then there's no other way for pressure release than the tube actually exploding. And we have sometimes incidences when people do this, when people store in the liquid nitrogen, and this is super dangerous. Also, you should reduce the exposure of vials to temperatures above nominal storage temperature. I mentioned this already earlier. Make sure that your picking times are very quickly, that you, before you pick a sample, you make your mind up about its location so that you find it very quickly. And you can even think about automated storage devices, because they only retrieve one sample that you need and everything else is kept at low storage temperatures. Last not least, consider the utilization of CE mark cryovials, because they are certified and approved for storage of in vitro diagnostic samples. On the right hand side, you see a very basic and simple trick how to store in the vapor phase. The metal rack, which contains the different racks and cryovials, stands on the bottom of the liquid nitrogen tank, where also the liquid nitrogen is. So just keep the lowest shelves empty. Don't put any racks and vials into these shelves, and then you are safe, and your samples will never be in the liquid nitrogen. In many workflows, there's actually a point in time when a sample needs to be shipped somewhere, to a different laboratory, to an external collaboration partner. And very often these people are not in the same country, and then air transportation of samples comes into the play. Make up your mind that there are regulations available for the air freight and shipment of biohazardous material. They involve some specifications about the primary container, secondary containers, soakable materials, and a rigid outer box with certain labeling. There is a lot of literature and regulation about this available in the internet. I want to draw your conclusion to the primary receptacle for this shipment, because in almost any case, in almost all the cases, it's identical with your cryovial, your primary storage container, because you will not thaw the sample and put it in a different container. Here it's important that your storage container and also your primary receptacle for shipment is actually IATA guideline approved, which means that it's about the tightness of the screw cap, the container, that all these tests have been carried out, which make sure that at reduced air pressure, which you usually see in airplanes, the tube is still not leaking, but it's leakage proof. So in this slide, there are more best practice tips, this time about thawing and reconstitution of cells. First of all, you should thaw your samples as quickly as possible. You should do this in a water bath at 37 degrees Celsius. You should agitate your samples during this step, but not submerge them completely into the water. Afterwards, you wipe the external with ethanol, and then you keep your samples at 4 degrees in an ice bath. And afterwards, you have to remove your cryoprotectant. For this purpose, you should transfer the tube's content to fresh growth media. You should quickly spin at around 100 G for 10 minutes. Exchange your media to fresh growth media. Do this very quickly. And you should also avoid any refreezing of thawed cells, which actually also applies for non-cellular sample specimens, because this usually kills cell viability, or it kills biomarkers in your serum or whatever type of sample you have. On the right hand side, you see a best practice tip in a picture. It's the idea that after removal of a cryovial from your storage, from your liquid nitrogen storage, you untighten the screw cap, you loosen it a tiny little bit, and if for some reason air pressure has built in or pressure has built in the tube, then this can be released and there will be no damage of your tube and sample. As mentioned earlier, the quality of your recovered cells should be assessed after thawing. There are several methodologies available here. And if we first of all look at bacteria, it's serial dilution and counting of colony forming units. What is the preferred methodology for assessing the amount of viable bacteria in your recovered stock? For mammalian cells, the first thing you have to do is a visual inspection. Trained people see if the cells are viable or not. You should combine this with a trypan blue dye exclusion test, which allows for the quantification of living cells in a hemocytometer or an automated cell counter. On the right hand side you see a picture, I guess many of you know this, where you see several cells which have excluded the dye. They are viable, and 3 cells were not capable of excluding the dye. These cells are not viable any longer. There's a phenomenon which we do not have the time to talk about here in detail, but I want to at least mention it. It is the cryopreservation induced delayed onset of cell death phenomenon. There's a piece of literature, a citation I provide here, which I recommend for further reading. And what is behind this phenomenon, in simple words, is the fact that when you thaw your cells, they may already have suffered and they may already have triggered a program of apoptosis or cell death, but you only see the effect of this later on. So the cells are later on really dying and showing very low viability, whereas when you assess their viability right after thawing, they still seem to be okay. And this is an important phenomenon because it may really interfere with the outcome of subsequent steps in your process. Last not least, stem cells. Here it's usually their differentiation potential or their clone forming capabilities which are assessed and which are criteria, parameters for their viability and applicability in downstream processing. With this, I want to come to the second part of my presentation about contamination, how to detect contaminations, what are their sources, and how to avoid them. Contaminants actually fall into different categories and they come from different origins. The first group is polymer additives. These polymer additives may leach from plastic labware into your sample. They may leach from the storage vessel into your sample and thus contaminate your sample. In order to exclude them, you may ask for a labware, you may ask your vendors for certification, which indicates that labware is made from very virgin medical grade polymers with low additive content. And you can also ask for certain certificates. There is, for instance, a very common thing. It's a free of phthalate ester statement. And then you can be sure that these substances, these additives are not contained in the labware in the cryostorage vial you are using. A different class of contaminants is biological contaminants. Endotoxins are included here, DNA, DNAs, RNAs, TSE and BSE. These contaminants can be derived from contaminated supplies and media and also from storage vessels, but also they can be introduced if improper aseptic techniques are used in the lab. It's just a bad practice which can lead to such contaminations. Of course, the ways of fighting such contaminants are clear. I guess it's asking for labware which is certified free of endotoxins, DNAs, DNA, RNAs, TSE and BSE, and also make sure that you apply best practice and best aseptic techniques in your lab. Another group of contaminants is also of biological origin, but here we talk about whole cells. It's microorganisms or cells, bacteria, molds, yeast, viruses, protozoa, mycoplasma, but also cross-contaminating cell lines are falling into this group. The origin can be very diverse. It can be non-sterile supplies and media. Again, it can be improper aseptic techniques, but it can also be the mixing of different cell lines. And you are actually thinking you work with a very clean cell line, but it's actually contaminated with a different one. The ways of fighting these contaminants here are use sterile labware. There are certain levels of certification available. CryoVials can be obtained today with a sterility assurance level and certification of 10⁻⁶. I personally suggest this should be gold standard today, and also supplies and media should be sterile. And last not least, again, it's good laboratory practice and proper aseptic techniques that have to be applied in order to assure that such contaminations are not occurring. On this slide, I show you an example of a quality certificate, and I also want to show you how to read through it. So a good quality certificate comes with a clear title. This is the quality certificate. It has references to a certain product code, to a shelf life, if applicable, to a sterility assurance level. These are the things we see in the bottom portion. And then all the different things that are tested for, if it's physical properties, if it's functional properties, or if it's contaminants or other things, are very clearly stated. There are often the methodologies provided in a good certificate. I expect this to be the case. And also detection limits are there. And these certificates are something you should ask for, you should read carefully, and then put them in your documentation and make sure that everything that is stated in the certificate is in accordance with the criteria of your workflow and your needs and demands. A very prominent example for biological contaminations is mycoplasma contaminations. Up to 35% of all continuous cell lines are today believed to be contaminated with mycoplasma, and potential sources are cross-contaminations from infected cultures, laboratory personnel, so human beings, and also contaminated culture reagents. Best practice tips here include the testing, the frequent testing of your cells for potential mycoplasma contaminations, utilization of DNA staining, PCR technologies, ELISA or microarray-based mycoplasma detection kits or similar methodologies. And it's also important, once more again here, that you do not submerge your vials into liquid nitrogen. We talked about the danger this bears in terms of cells, of tubes breaking and exploding, but also liquid nitrogen in these liquid nitrogen tanks is very often contaminated with mycoplasma. They stay viable in the liquid nitrogen, and if you submerge the tubes and liquid nitrogen is entering, you have a very rich source for mycoplasma contamination. On the right-hand side, there's an example of such a detection kit. It's the CytoInspect mycoplasma detection kit from CryoPreservation. It involves the steps of a PCR, of a microarray hybridization step with washing, then there's a scanning step, and you quickly have your results. You can detect 40 different mycoplasma species, and you can also differentiate them, which often is a very good indication where actually your contamination is coming from, because the different strains usually come from different sources of contamination. There's a sensitivity level of less than 10 CFUs per milliliter, and it is validated after ICH Q2R1. As mentioned earlier, also contaminated cell lines are a source of error. Today we believe that 15 to 30% of cell lines are not representative of what they are believed to be. HeLa cells are a very common contaminating cell line, and in the center table of this slide you actually see several cell types like HEP2, L41, L132 and WRL68, which were all believed to be of a very specific origin, like larynx, like bone marrow, like human embryonic lung or human embryonic hepatic origin. But actually all of them were later on identified to be HeLa cells. And you can imagine the shocking moment when this is identified, and researchers have already published their results and have interpreted them in a completely different and wrong direction, because they thought they were dealing with a very specific cell type. Best practice tips here are: you should never work with two different cell lines in the hood at the same time, and you should confirm the identity of cells you obtain from other labs and also which are kept in your stock by either isoenzyme analysis, by karyotyping, by DNA fingerprint, or by marker detection. In the last part of my presentation, I want to finally talk about high-throughput cryostorage, modern options for tracking and automation of cryopreservation tasks. On this slide, I want to give you several basic examples for good practices with regard to freezing, documentation, and also tracking samples. On the left-hand side, you see a freeze data form These freeze data forms allow for writing down everything around the freezing protocol, the freezing process, and later on the recovery and also assessment of quality and viability of your samples and your cells. So what you achieve with these forms is that you can correlate all the specific data of your protocol, your freezing protocol, with how viable were the cells afterwards. And in other words, this allows for optimizing your freezing protocols towards a better recovery and viability. In the center, we see a cryo inventory protocol. They can be very simple. And the concept here is if you write down the position of your samples in your cryo box and in your freezer, you can choose your sample. You can find the position of your sample of interest before you open the freezer door. And this reduces the time the box is taken out and all the unneeded samples are exposed to higher temperatures dramatically. And this also at the end of the day will increase the quality and viability of your cell stock a lot. And to the right-hand side, we see an example, a screenshot of a sample management software. Such software allows for the entrance of all kinds of sample-related information in a software format. They are today available at good, decent entrance levels in terms of price. Such softwares, and what they also allow for is that you can use barcoded tubes and you can scan the unique identifier encrypted in the barcode of the tube and introduce it in your sample management software, and then start with the typing in of all the related information. And with this, there's no need anymore for labeling your vials, for writing on writing areas, or for putting adhesive labels on the vials. You just purchase the barcoded tubes, you use the software, you use the scanner, and you are done, and you have an unambiguous process of sample identification. So in this slide, you see a similar workflow as shown before, but this time adapted with regard to biobanking and specific needs of high throughput cryopreservation tasks. Everything starts with preparation of samples again, but the need for standardization and reproducibility at this stage is much, much higher. Also, the time from sample drawing to freezing should be short. It should be less or equal to 30 minutes, which is really challenging. During storage and transport, there's often the need to anonymize samples. There must be unique identifiers on the samples, which allow for secure sample tracking and secure link to all the sample-related data in a sample management software. And also the requirement for maintaining sample integrity is very high here. Until analysis, the cold chain should be maintained from freezing to analysis in order to allow for good results after such a cryopreservation task. There are several solutions available for biobanking and high-throughput cryopreservation tasks. They include labware, they include machines and devices, lab automation, and they include software. Let's start with the labware. Here the trick is that the labware, the racks for the cryovials, are following the SLIS ANSI standard for microtiter plates. And by doing so, actually the labware can be used with all these automated liquid handling systems, decappers, with all these nucleic acid preparation devices and so on, which work with standardized microtiter plates. Another feature of labware is that the tubes and the racks usually are endowed with machine-readable barcodes. I mentioned them already earlier. And these barcodes allow for the machine-readable identification of their unique identifiers. Everything can then be transported into laboratory information management software. And so at any time, these tubes in the process can be identified, and it can be recorded what happened to them and how they were processed. And I already mentioned it, that laboratory information management systems are often at the core of everything. These are software programs which allow for the integration of tube identifiers, rack identifiers with processing data, with storage data, and even with analysis results at the very end of the workflow. Here you see an example for such a barcoded cryovial. The vial has a unique identifier. In this example, it starts with the letter U and there are digits following. This identifier is unique. You will find no tube with the same identifier in your cryostore. The identifier is represented in three different forms. one time as a human readable feature. You can just read it with your bare eye. You don't need any barcode scanners. And this is good whenever you need to identify a sample and maybe cross-check it versus some shipping documentation. On both the tube and the documentation, there appears the same identifier, and then you can just read all the sample-related information in the documentation. But there's also a representation in form of a linear barcode. Here you can use a low-cost barcode scanner to retrieve the information. It's the same ID, and just introduce it into an Excel spreadsheet or any kind of software with a very low-cost option and without the risk of any human-introduced error. And also, the unique identifier is represented in the form of a data matrix code. Here, the idea is that the same identifier, again, can be presented together with other tubes identifiers in an array, and here bulk readability comes into the play. So you can read an entire rec, often it's 48-way rec, so you can read 48 identifiers at once. You get also positional information, so you know which tube with which identifier was in which rec position. So you get positional information, you can relate it to rec IDs, and you can relate it to individual IDs of individual samples. So these barcodes, these machine-readable codes, finally now allow for a lot of advantages in terms of tracking and traceability of your samples. There are many different concepts available, but I want to show you one example how such an integration could look like. You could start with recs, where certain tubes are introduced in the recs. And then you do a bulk scanning. You scan at the bottom of the rec, and the data matrix codes are actually shining through because there are bottom holes in the rec. And then you read the individual IDs of the tubes, and you see their position. You relate it to the rec ID. Everything is introduced into your sample management software, or in the most simple case, into your Excel spreadsheet. And then you can just start to type in all kinds of sample-related information. Afterwards, you just search your database, your software for individual tubes with certain content. You get their IDs, you get their position, you retrieve them, again, very quickly without other samples suffering from exposure to higher temperatures, because everything is very quick. And then you do a quick cross-check, either with your bare eye or with a handheld scanner, and make sure you retrieve the right tube with the right ID and you work with it. And in this cycle, you can always use empty positions which became available for fresh tubes later on. So tubes go in, tubes go out, and again, you take all the advantages of the barcoding, which allow for very secure and very straightforward sample tracking. So with this, I want to end my presentation. I thank you very much for listening. Since this is not a live presentation, but a recorded session, I want to draw your attention to our email address that we have available, webinars@gbo.com. Please send your questions to this email address or any other kind of request around this webinar here. And then our experts will take care of your questions and you will be answered promptly. Thank you very much and have a good day.